It's a pleasure for me to be here at the symposium in honor of Brad. He's absolutely one of my heroes in statistics, and that is attributable not only to his genius, but his kindness as well as a person. So thanks, Brad, for all of the contributions that you've made. In fact, I did not a very thorough, but just a quick Google search on fMRI and the bootstrap, and there were over 600,000 matches to that. And I'm sure if I did something in depth and actually did site tracking or something like that, that it would be, the impact would be even larger. So your contributions are, I think, felt many times over in many different areas. So I'm going to be talking today about just basically the analysis of large-scale neuroimaging data. We have had some introduction from Brian Caffo, so I'll build on some of the ideas that he presented. In particular, we'll make some comments about inference and other objectives. So I'll start out just briefly by giving some background, and then I'll talk about, we've spent a lot of time, as Gina mentioned, dealing with aspects of spatial correlations present in neuroimaging data, and I'll talk about, it's not our most recent work, but it's one we have software for, so I'll talk about that, and I think it's one that is easy to follow and understand conceptually what we're trying to do, and then we'll briefly mention some other projects as well. So for those of you who are not familiar with fMRI data, we bring subjects in and acquire scans on them. The scans are usually taken either at what's called resting state, where essentially you're left to think for yourself while you're in the scanner, so you're not explicitly told to do anything. You can imagine being in a very big machine that makes loud knocking noises, that there's still a lot of neural processing going on that is hardly like rest, but nonetheless we term it as resting state. Alternatively, we ask subjects to engage in a task, and the task may be to respond to a set of questions that involve some kind of cognitive function, showing pictures that evoke an emotional response or memory task. In fact, I'll talk a little bit later about a specific study involving a memory task. So there are various things that you can do. There is a restriction, an obvious restriction, that you can't do things in the scanner that require a lot of movement, because the movement distorts the quality of the images. So most of the tasks have to be presented via PowerPoint slides or something like that, and involve button presses at the most. So if we say acquire, is there a laser pointer? No, okay. I'll just use the mouse, it'll do just as good. So if we acquire, say, 6 scans on an individual, to give you an idea of sort of the characteristics of the data, we take each scan and string it out into a long column vector like this, and then we cut across any particular row of that. What that gives us is the neuroactivity as a time series, just the evolution of brain activity at a particular location. Thank you very much. So again, this is just a time series at one location in the brain. So, and the number of voxels, those volume elements, tends to be very, very large, in the tens of thousands, maybe hundreds of thousands. So essentially the data on one subject can be viewed as hundreds of thousands of time series. And then naturally we use the images to construct the columns in the first place. So if we cut down a column, it just gives us an image that's 3 dimensions, and we can view it as 3 orthogonal cross sections. All right, so now to describe a specific study that involves working memory in patients with schizophrenia. Schizophrenia is something that heavily impacts working memory, and more specifically, or more generally, I should say, executive functioning, which involves planning and things like that in the brain. And so this particular study sought to look at neuroprocessing underlying related to working memory tasks in patients with schizophrenia. And there were 28 subjects, 15 patients, 13 healthy controls. The working memory task was in the form of a serial item recognition paradigm. And it went as follows. So there was an encoding set where numbers were shown, single digits were shown in red, and there was either 1 digit, 3 digits, or 5 digits, and that was shown momentarily. And then after a brief delay, a blank screen, probing set digits were shown. And subjects were instructed to respond with a button press using their index finger if the probing digit matched one of the digits in the encoding set, and their middle finger otherwise. So naturally, in the 1 digit case for the encoding set, it's quite simple. You're shown a single digit, and then subsequently you're shown a series of digits, and you just have to click if it's a match and not. But the working memory involved in, say, the 5-digit case is increased because now you have a brief look at 5 digits, and then after a period, you're trying to restore those in working memory and then respond with the appropriate button press on whether or not the probing digit was included or not. Okay? So for each one of the cases were run separately where we have 1, 3, or 5 digits, and so for each one of them there was 177 scans, and subjects were brought back in for rescanning on a different day. Okay, so in total, there were over 1,000 scans per subject. And the objective that will be considered here is to compare the working memory-related brain activity between the schizophrenia patients and the healthy controls. Now, and I'll say a little bit more about how we quantify that. All right, so just a summary of some of the characteristics of the data. There are at the time of analysis, again, after the pre-processing, there are roughly 900,000, in fact, over 900,000 voxels. Not all of them fall in the brain. You can do smart things like throw away things that are not included in the brain to help solve some of the computing demands, but still that would cut it down to maybe 300,000 or so unless you restricted it to regions of interest. Again, the number of scans is usually in the hundreds for fMRI, and in the case of the working memory task, we have 1,000 scans, including the multiple days or multiple sessions. So almost 1 billion spatial temporal data points for each subject, and 26 billion in that study for all subjects. So the point of this is I think that your entry into the analysis of fMRI data should be from a simple perspective and then progressively try to relax assumptions and to get more sophisticated. But otherwise, you run quickly into computational bottlenecks. The temporal correlations that are naturally apparent are two. You have scan-to-scan correlations. The scans are acquired roughly 2 to 3 seconds apart, and that's why we get that time series. And so things like autoregressive model have been applied to address those. And then we have these repeated measures types of correlations by design, the fact that we have a scanning run under one condition, a scanning run under another condition, a scanning run under another condition, and then a day break, and we repeat those. So we'll get some, those are similar to what you might have in other areas of statistics with longitudinal data. So we have another layer to the temporal correlations there. The spatial correlations are quite complex to tackle. And I'll elaborate on this in a moment. You have correlations between neighboring voxels, almost always true. Yet you have very strong long-range correlations. There's very intricate wiring in the brain that connects things that are distant, and as a consequence, you get firing all over the place. The visual system is one good example where a lot of the visual processing is done in the back of the brain, but you have long bundles of axonal fibers that connect to other areas of the brain for processing. So you get distant voxels that are highly correlated, which is not true in many areas of spatial statistics. And then just the mere number of voxel pairs. There are over 400 billion voxel pairs. So again, the point is that you have to somehow try to simplify this into a reasonable structure. You're not going to capture everything, but try to capture some of the main correlations in the data at the time of analysis. All right, so just to elaborate on the spatial correlation a bit, what this image does, this functional image on the left, calculates what I refer to as a functional distance. Essentially, these, in fact, I generated this figure from the schizophrenia data, since this is what I'll be working throughout. So essentially, you can imagine a vector of 3 elements, one as a mean of the 1-digit task, one as the mean of the 3-digit task, the third element being the mean of the 5-digit task. And so every voxel has this 3-element vector. And you can calculate any measure of dissimilarity standardized to 1 here of the distance, let's say, between that particular voxel, and then the color represents the distance for every other voxel in the image. Okay, so blue here is low, which means highly similar, and in the neighborhood of this crosshair, we see a lot of blue. Okay, and that's very, very typical if you look around and explore these images. You go off in a fairly short direction, and things are highly dissimilar there. But then look in the front of the brain, right? Again, we have this blue that shows a high degree of similarity. So if we were to use a standard spatial model that says that the spatial correlation should decrease with increasing distance, then we'd get something that looks like this. Okay, so the fact that these maps do not match means that we have to do something beyond sort of using concepts of geometric distance in terms of modeling. All right, so 3 common objectives in neuroimaging, functional neuroimaging analysis are what's generally referred to as activation studies, where you're looking to determine brain regions that become more active when performing a task than not, and then in that same light to be able to perform group comparisons or comparisons before and after treatment with respect to those changes in activity. But those are all localized. So at this particular point in the brain, is there a difference in neuroactivity between the 5-digit working memory load and the 1-digit, or between patients and controls? And then connectivity studies that Brian talked some about, you try to identify what brain areas show similar patterns of activity over time. And this is more sort of driving at networks. You're trying to identify networks in the brain that seem to work in synergy somehow when you're either at rest or involved in a task. And then lastly, again, Brian talked about this, is prediction and classification is emerging as a very important area in the field with, I think, promise for potential clinical applications if many of the methods prove valuable. I'm going to focus, actually I'll say something about all 3 of those, but in the beginning I'm going to focus on the activation studies paradigm, and a common analysis framework there, just to give you an idea, it proceeds in 2 stages. So the first level is a huge data dimension step where essentially you consider data on subject by subject basis. You have this long time series, and what you try to do is just estimate regression parameters that correspond to the task. So we'll take that time series of, I guess, within a run we have, so let's say 500 or so time series if we wanted to keep the parameters from days separate, or 1,000 if we wanted to combine them. And then we reduce that down to 3 elements: a mean for working memory load number 1, a mean for working memory load 3, and a mean for working memory load 5. And then at a second stage, what we try to do is take those individual specific parameters that we now use as data at the second level and then model them to get group estimates. So that's the basic construct. At the first level, you obviously have to deal with the fact that you have a time series. Those aren't independent observations. And so, again, autoregressive modeling ideas have been used a lot to address that level of the correlation. After you've obtained group estimates at each and every voxel, then if you want to proceed to inference, you calculate t-statistics and then threshold somehow. There's an obvious multiplicity issue here. You have hundreds of thousands of voxels, and you're trying to declare things that's statistically significant or not. What does that do to your family-wise error rate or to the number of false discoveries? And so I say Bonferroni type adjustments have been made. Those are usually with respect to some defined cluster size or just some arbitrary divisor that you use, because if you use even 100,000, you won't find anything significant. And then FDR has been used a lot. In fact, I did the same quick experiment with fMRI and FDR, and there were 400,000 matches for that in Google. So that's penetrated the literature quite a bit. And then random field theory is another, where you don't take into account any spatial correlations, just as I described, but then you view the image as sort of this three-dimensional statistic. So you have to now have a field that you can use for thresholding there. So two things that come to mind. One is that the two-stage approach, well, we can do that in one stage because we do it in other areas in one stage. So this is, in a sense, taking a step backward in the field of statistics. There's very good reasons for doing it in neuroimaging, and the biggest reason is computational. So you do lose some efficiency, but even running just a mixed model on a voxel by voxel basis, you have to wait a really long time to get results. So this two-stage approach emulates a random effects model, and computationally it's a lot easier to do. The approach that I just talked about, where you basically fit a model in every voxel separately, assumes independence between different brain locations, and so by doing so, it targets this activation analysis objectives but disregards functional connectivity. So what we wanted to do is firstly to try to borrow strength somehow. We know that there are spatial correlations present in the data. How can we try to capture those and borrow strength? Secondly, if we capture them, they relate in some way to this notion of functional connectivity. So the second idea was to sort of unify this activation paradigm and a specific version or flavor of functional connectivity. And we've done several stabs at it, and again, what I'm showing is not our most recent work, but in fact, Brian Caffo was involved on this work as well, where what we decided to do was use neuroanatomic information, and we do that to break the brain down into regions, where then the regions will serve as sort of natural units within which to borrow strength, and then between which we can borrow strength by modeling the spatial correlations. There are several choices for templates that you can use. We use things that are standard in the fMRI literature, like Brodmann areas or AAL. Arguably, these are too large, so some subdivision of these may be important. And again, we're going to get spatial correlations within and between. We still, we don't remove the voxel level analysis, so that's there. The boundaries brought about by the regions essentially allow us to capture spatial correlations, but then also, if we want to look at regional level effects, we have kind of a natural way to do it, and we've incorporated some of the spatial correlation that's important for addressing those. All right, so real quickly, here's the basic model. It excludes covariates and things like that, which are that are also a part of it, but to simplify notation, those are not included here. So subject I, or subjects are indexed by I. G represents the particular brain region, and this is a vector with all of the voxels in that region. Okay, and so this is just modeled in terms of a group level mean mu, so that's essentially what we want to do inference on. And then we include a random effect here for the I subject in the G brain region. Conditional on that random effect, we assume independence, but this conditional structure then allows us to have an exchangeable correlation structure within each brain region. which is a very strong assumption, I should say. So we have these brain regions, and every pair of brain regions, every pair of voxels within that brain region is assumed to have equal correlation. So we've actually modified that in more recent work, but that was the assumption here in this first piece. These random effects, what we do is concatenate them. Here they are. So we have, again, one random effect for each region, and so if we just stack them up for all of the G brain regions, then we assume that that follows a multivariate normal distribution with mean 0 and covariance matrix gamma. So the off-diagonal elements of gamma capture covariances between the random effects from different regions, which, and then you sort of work out the algebra, then brings about correlations between voxels that fall in different regions. So that's the basic setup. I'm not going to go through all of the prior selections, but we just wanted to point out how this model tries to tie in the correlations in the data. I will say that we use, at least with our software, we try to use minimally informative priors. If you have information that's available to you that should cause you to use more informative priors, you certainly can do so. But otherwise, by default, we use the objective Bayes, as Brad phrased it, approach. So just to briefly show you the software, if any of you are interested in using the software or downloading some free data and trying it out. I'm not going to go through this in detail, but this shows you the basic interface. And essentially, you can read in data from various types of imaging file formats, or if you've already read them in, the reading in itself takes some time. So if you have already read them in, you can just load in the previously loaded data. And then you set your contrast of interest. So here what we did, and this will be important later when I talk about activations, what we wanted to do is compare, again, schizophrenia patients and healthy controls. So we contrasted the working memory level, the 5-digit working memory load versus the 1-digit for each group, and then took the difference between those groups. So when I say the difference between the schizophrenia patients and the healthy controls, it's with respect to that difference between the 5-digit and the 1-digit working memory load. Very often, you may be interested in just a subset of the regions, and so this panel allows you to select regions. And then we also impose a sample size constraint. If you have 3 subjects, we don't want to try to compute several hundred pairs of correlations between pairs of brain regions. So we restrict, in some cases, the number of regions included in the analysis if you have a limited sample size. But otherwise, if you have a sufficient amount of data, you can include all the regions, and the computations don't increase that much in terms of time. These are, again, the default choices, the objective Bayes approach, but you can tailor those. And then you estimate and plot. So when you estimate the data, you'll have lots of just what I call basic plots that allow you to explore, make sure things seem to be working well. We do as good of a job as we can in terms of diagnostics, convergence diagnostics. So things like the region level parameters are very easy to do, but you also have the voxel level parameters. So we have some things that are computed, but you can't actually go in and look at trace plots for 100,000 voxels. And so after looking at those, you can do a little bit more anatomically informative graphics, and this shows one of them. It's just sort of a dynamic graph. As you click around, you can see different areas that show increased probability of activation, where activation is defined as I mentioned previously. So this is a typical map that you'll see in neuroimaging experiments. However, I think the interpretation here is a lot more natural, that these hotspots represent the probability of activation from the posterior probability distribution, and you can threshold wherever you like. And so from this, you see the putamen is one area that has been involved in or implicated in memory tasks. You actually get, this is the superior frontal region, but as are these. So that's a really huge area, and as you get toward this posterior range of the superior frontal, it reflects motor. So the fact that you're doing button presses, for some reason there's a difference in activation. We wouldn't necessarily expect that. We would expect that it cancels out, but there's some highly probable difference in activation related to that. You can look at regional effects as well. I'm not going to go through all of the functionality, but another thing is we can look at what we call task-related connectivity maps, and the task-related connectivity comes from the fact that these are correlations. You can think of just a covariance matrix between all of the regions, and so these capture correlations operating at the level of the summary statistics rather than correlations between the original time series between these regions. And the latter description is, I think, what many people in neuroimaging think about as functional connectivity. So we preface it with task-related connectivity or paradigm-related connectivity to try to distinguish that. So this just shows a map. You can pick any group, and so this is the working memory load 5 for a schizophrenia patient, healthy control, same condition. And so just by flipping back and forth, you see that there's increased connectivity for the schizophrenia group relative to the control, possibly suggesting that they have to recruit other brain regions in performing these tasks and don't process things quite as efficiently as the healthy controls. Okay, so just to quickly summarize some of the features of this, what we call BSMAC, it's the Bayesian Spatial Model for Activation and Connectivity. Considers activation objectives and task-related functional connectivity. 5 minutes. Okay. Thank you very much. It yields the easily interpretable posterior probabilities of activation, and then also it permits voxel and region-level analysis or inferences. Does not account for temporal dependence between multiple sessions, and that became important later because we were involved in a task where a study where the investigators wanted to do that, and actually another case we were involved in a study where we wanted to try to predict future neural activity, and I'll say a little bit more about that in just a few moments. But to do that, you then naturally have to build in that second layer of correlation between the sessions to do it. And then there's a fairly simple intra-regional correlation model, which I mentioned that we've extended that in some other work as well. So I'll quickly, in my remaining few minutes, just talk about some other things. So prediction studies, again, it's an emerging direction in neuroimaging. High promise to increase the clinical applicability of fMRI. MRI, just looking at structure, is very important for looking at things like neurological diseases that cause lesions in the brain, or if you have a stroke, you're able to visualize that. So the clinical applicability of MRI, I think, is at this stage much higher, but there's hope for fMRI to increase in the future. So we have a study that we're involved in where the ultimate goal is to try to predict treatment response. So on the basis of not just imaging data, and after Brian's talk, I'm glad that we have more than just imaging data. But we also have lots of genetics information and things like that. And so on the basis of all of this information, can we try to say who's going to respond to treatment and who's not among patients with major depressive disorder? As an intermediate objective, we wanted to see, well, all right, we've had these longitudinal scans. Can we use earlier scans to predict later scans? And so we developed a spatial modeling framework that extends what I just presented to do that. And briefly, two extensions. One is that we sort of use a more focal neighborhood rather than the entire Brodmann or AAL region. We look at just the immediate neighbors, and then again build in correlations between the pre-treatment and post-treatment. This could be session 1 and session 2 if it's not a treatment study, but in our case it was pre and post-treatment. So just to quickly show you some images of how we're able to predict. This is observed patterns of neuroactivity. This is predicted patterns of neuroactivity. And if you examine it, you'll see some differences. But on a whole, we do fairly well. This is just another subject that shows a different pattern. If you use just a regular general linear model, like a standard approach, you have the same map for every subject. But our map is sort of a compromise between group level estimates and individual level data. And so you see here that we still for a quite different pattern from what we saw earlier, we still managed to predict fairly well. Okay, the last thing I'll just comment on, we're also interested in looking at different types of imaging data and fusing the imaging data together, if you will, to hopefully to result in a more informative analysis. And one such example is the use of diffusion tensor imaging, which gives us information on the integrity of white matter fiber tracts in the brain. And so if we're thinking about connectivity and we have functional associations between different brain regions, how does that arise? It could be on the basis of a direct axonal link between two regions, but it doesn't necessarily have to be. So we've actually begun to do some more modeling of joint estimation of diffusion of structural and functional connectivity. But to begin, we wanted to just do some descriptive things. And so the idea is that we can look at functional data, so the time series in 86 regions, and on the basis of that compute a measure of similarity or dissimilarity, and then similarly using diffusion tensor tractography, look at connections or not between different regions, and then blend these together to form some metric that can be used to define functional networks. And so we've been able to do that pretty successfully. This shows just the results of 3 clusters of brain regions that emerge from doing that. And these are time series for one subject for all of the regions together, and essentially these are partitioned into what you see here. These, in fact, are not the most coherent regions. They are regions that are involved in the default mode network, which is important for resting state data. And so that's why I selected those. There are other regions where you have highly coherent activity between the regions, and in fact, one of them is the olfactory system. It's a very distinct pattern that you get. You could, across subjects, you could look at the clusters and you could pick which one contain the olfactory regions based on the patterns of activity. So that's just another example. So in summary, I think I'm about out of time. Neuroimaging applications involve the analysis of massive data sets, and to be practical, I think there should be a focus on computationally simple methods, and I say that in a relative sense because even the most basic computations can be quite involved when you have so much data. Complex spatial and temporal correlation structures exist, and I think we want to explore that because it's not just for the sake of borrowing information, but there might actually be inferences that are drawn or insights that are drawn from estimating those parameters that are important. In fact, for many disease processes, it's the correlation between regions that is important, more important even than the localized function. So there's good reason for doing that. Multiple testing approaches are very well suited for the field, and it's very important for problems or there are very important problems that do not involve statistical inference as well, but it's important for statisticians to be involved in them, like the prediction or the partitioning methods, just to still have basic principled ideas. When I find myself working in this area with people, very, very smart people from many different fields, I find what statisticians often bring to the table is just a principled way of approaching things and hopefully leading to being able to quantify inference. And just quickly, I'd like to thank some members of our group who contributed. They're members of our center, our Imaging Statistics Center at Emory. And then to conclude there, I'd like to ask a question from people sitting in the front row. So that leaves 2 of you. Do you recognize the setting here? Okay. So it's just on the back side of the oval at Stanford. I was there this June. My sister finished a law degree and jointly a PhD, and so these are my 2 boys who right near the department. So, thanks.